Good day. My name is Dr. Hlumani Ndlovu. I work at the University of Cape Town as a senior lecturer. So I was born in KwaZulu-Natal, specifically in the city of Pietermaritzburg. So just a few kilometers from town. So I was born in a village, grew up basically, typically like a village boy, go to school, come back from school, will then go and, fetch the cows from the fields. That was very, tough. You learned to take responsibility at a very young age because if the cows don't come back home, then usually there are consequences for us back then. So I went to the local schools in the village until, I finished matric. After I finished matric, I got a couple of distinctions, but then I had not really applied. I had not thought about university deeply. So when I got my results, this is when it dawned that, hey, actually I could go to university. And I did not know what to do. And luckily, there was a guy in the community who had gone to the University of KwaZulu-Natal. Back then, when I finished matric, it used to be called the University of Natal before the merger with the University of Durban Westville. So we went to him to say, here, I'm a Max, how can you assist? Luckily, the guy was in the SRC, so he knew the right doors to go and knock. And we went to the university with him, and I presented my marks. Then and there, I had to apply via the CAO, which is the Central Applications Office, which basically is able to take your application and deliver it to all the universities in KwaZulu-Natal specifically. So we did that on the same day. I applied for NSAS also on the same day. And then I waited to hear the outcomes, and luckily I was admitted to the university. Now this happened back then because I think the demand for universities was not as high as now. Now if we're to do or take the same approach today, you will not get into university because the demand is so huge. Hence why now as a grade 10, grade 11 learner, you need to think about matric, you need to think about university, and kind of have an idea what route you want to take ultimately when you get to the stage. So that you can then have, one is you can work on your marks, because your grade 11 marks are critical in influencing your admission to university. And we saw this particularly this year with the admission of students for 2021, because most universities last year could not do the NPTs, the national benchmarking for admission of students to university. Now, when they did not do the NPTs, that means that then they will then use only your grade 11 mark to make an offer of acceptance into first year. So the grade 11 marks are critical in giving you an opportunity to be looked at by the top universities in the country. And that's why I want to say now, don't be like me back then when things were, maybe I would say, easier. Because now the competition is way too high. You need to plan ahead of time, work on your marks, make sure that your grade 11 marks are good so that you have a good chance of getting into university. Now, as I said earlier, I went to study at the University of KwaZulu-Natal. This is where I did a degree in what is called biomolecular technology. So biomolecular technology basically is a 3 major degree. So it combines microbiology, it combines biochemistry, and genetics. So you get exposed to these 3 different fields in one degree. So it's a very nice degree in that sense. It's a broad degree. It's able to give you multiple avenues to follow for your postgraduates. Or even when you're looking for a job, I think you can go into different fields with that kind of degree. And after I finished my degree with a distinction from UKZN, I stayed on and pursued my postgraduate studies. So I did an honors degree, and then after that I did a master's degree. And I finished both of them with distinction again. And what I was doing for my honors and masters, because I did the same project for both honors and masters, was I was looking at, trying to look at parasites, or working on parasites actually, that cause what is called sleeping sickness. So this is a disease to find mainly in cattle in Africa. So it's a huge problem. It causes huge economic losses for the continent. So we were trying to understand, or express these proteins that we thought could have a potential as a vaccine for this particular, parasitic disease. So we were specifically working on proteins that are called, proteases. So these are proteins basically that will, or enzymes, that digest other proteins in the main. Now we're trying to express this protein in a native form so that we can use it in a vaccine platform. So I worked on that with my colleagues until I finished my master's. And then, of course, I'd spent by that time almost 5-6 years at UKZN. So I felt that I could no longer grow in the space, and I wanted a different challenge, a different space, so I can, expand and grow as a scientist. And this is when then I decided to move to UCT in 2009 to come to UCT and do my PhD. And I did my PhD again, continuing on the same vein, working with parasitic disease, but now working specifically on a disease called schistosomiasis. Schistosomiasis basically is also known as bilharzia. So usually what happens with this disease is that, you could just go and swim in a river, and these worms will just go through your skin. Like what you normally feel, you just feel an itching sensation, and worms just penetrate your skin and they go into your tissues and then cause havoc. So our interest in this context was to understand the immune response to these worms. So there are two stages, I would say, to the disease. The first part is when you have, predominantly the worms that are driving the immune response. But then over time, worms mature and they start producing these eggs. And this is the eggs that basically cause a lot of the symptoms that you see in humans. So the eggs get trapped in the liver. And in patients with severe disease, sometimes they'll even cross and get into the brain. They'll go into the intestines. So they literally just spread all over the body. If you have a high degree of infection without any treatment. So we're trying to understand then the immune response mainly to the eggs, and see if we can develop, drugs that could target, the inflammation that we see in the tissues. And I worked specifically, and we were using mice as a model of disease because, of course, you can't just go and infect humans. It's not ethically allowed. Of course, you can get samples from people who are already infected, but you can't just say, well, for a study, I'm going to come and infect people. Don't do that. So we then chose to use animal models, specifically mice, amakundana ngesizulu, to do this kind of research. So we'll infect mice, we'll then collect the different tissues, and then analyze the immune response to understand exactly, how do our bodies respond to this parasitic infection. So that's what I did for my PhD. And now, in my own lab, because now, I did PhD, I finished, then I did 4 years of postdoc. So postdoc is a stage after PhD, but before you get employed, if you want to be an academic. This is also just to advance your knowledge in the field as a scientist. So it depends on, of course, from person to person, but in most institutions, we have 5 years to do postdoc. So you can imagine that you spend 5 years, you spend 3 years doing your basic degree, right? Your undergraduate degree. Then you do 1 year for honors. Then you do plus or minus 2 years for masters. So that's already 6 years. Then you do another 3 years 4 years for a PhD. So you can say 10 years. So you already spent 10 years at university after you finish matric. And then you then do, 2 to 5 years of postdoc. So in total, really, you have invested 15 years of your life in this, career that you're trying to build. So it requires a lot of dedication, it requires a lot of hard work, it requires a lot of focus for you to be able to remain steadfast and focused on the work that you need to do, whilst you are still more or less a student, you are underpaid and overworked. And this is a reality. So it requires a person who has a clear drive and ambition to want to make it as a scientist. So you spend those years, and then at the end, then you look for opportunities, either to be a lecturer or to go into industry or to go into government. So then there are multiple avenues one can take after they've done their PhD and they've done their postdoc. Fortunately for me, after 4 years of postdoc, I was able to get a position at UCT as a lecturer. And of course, as a lecturer, you do many things. One of them, which is obvious, is that you do the teaching. So I teach medical students because we are based in the Faculty of Health Sciences at UCT. So I teach first-year medical students I teach them medical biochemistry. And then also I get to supervise students. These are students who are doing their honors, masters, and PhDs. I get to train them now to become me, right? It's a transfer of knowledge. This is what I do. And so this is really the bulk of the work that we do. We do the teaching of the undergraduates, but then the bulk of the work we do is research. You know, and I have multiple interests in research. So, as I said earlier, I do some work on worms or parasites, and I still continue to do that work even today. Because I mean, I think, you know, if you look at, for instance, with let's say, with how many people have TB, you know, in South Africa, and then you look at how many people have, you know, worms infections, the numbers are just so different. Like we have so many people who have parasitic infections in the continent, yet there's not much investment, you know, done or even research done to try and find solutions to those problems. Hence why I continue to do this work on typically what they call these, infections is they call them neglected tropical disease, because no one really pays attention to them. Hence why I find them fascinating to study in the lab. But also then I do some work on TB or tuberculosis. Now you know that tuberculosis is caused by a bacteria. So this is a small microorganism that you can only see under a microscope using specific chemicals to stain for the bacteria. Then you can see it under the microscope. It's caused by the bacteria called Mycobacterium tuberculosis. So what usually happens is that a person with TB will cough, you get these aerosols being released into the air, and then then another person will then inhale these aerosols containing the bacteria. Now these bacteria go into the lungs, which will get into what is called your alveolar spaces. Once the bacteria is in the alveolar spaces, in the lungs, then the cells of the immune system then come and attack. Because what is happening here is that they will know that something foreign that should not be in the lungs is in the lungs. Then they want to come and get rid of this bacteria. So that when they come, they then eat this bacteria, internalize it into the cell, and then try and kill it. More often than not, in people who have a competent immune system, so these are people who are healthy and they have a well-functioning immune system, they're able to kill the bacteria. So you take it in, you kill it, you are fine. In some people, the bacteria can remain almost dormant. So although some bacteria are killed, some bacteria will survive. And we call this a latent infection. So these are people who have no symptoms of TB, but they do have a bacteria sitting in their lungs. And the bacteria is not causing them any symptoms, they're not sick, they're not doing anything. So they have a latent infection. And then in some people, the bacteria will become active, and then they will have an infection. And then these are the people then who develop symptoms of disease. Like for instance, if you have TB, you know that you have coughs, you have night sweats, you lose weight and all those things. So as indicators that you have a TB disease. Now there are drugs available to treat TB and they work, but then they take a long time. I think you need to be on therapy for 6 months. So some people don't stick to the rules and take their drugs every day for 6 months. Some of them, they will take them for 2 days and stop, for 2 weeks and stop, for a number of reasons. And then this creates a space where the bacteria can find time to then mutate, to change basically. Because it should not be exposed to the drug, so you should know that, hey, this drug is coming to kill me. How can I resist this drug? And then it will mutate, specific regions in the DNA of the bacteria to allow it to now survive the attack from the drugs, right? And this is critical, and this is what is called drug resistance. So for TB, we have what is called MDR-TB, which is multi-drug resistant TB, meaning that it's resistant to multiple drugs. And then we have XDR-TB, which is extremely drug resistant TB, which means that basically now it is resistant to a lot more drugs, right? Like for instance, with MDR, usually by definition, it should be resistant to the first line drugs, and then we define that as maybe MDR. Now, when the bacteria develops resistance to MDR drugs, so there are drugs we use to treat MDR-TB, right? When it develops resistance to those drugs, then it becomes extremely drug-resistant TB. And the collateral damage of all of this is, as the bacteria becomes resistant and as the bacteria thrives in the lungs, what happens is that the lungs get destroyed, right? So like for instance, is if you were to take the lungs of a healthy person and compare them to the lungs of a person with active TB, you'll see clear differences. Where for instance, some of the tissue in the diseased lung is dead. Like you see this gray cheese-like material in the lung. And we call that calciums. So this is basically cellular material that has died. Now, in the lab, we are trying to understand what drives these, changes in the tissue, and how can we prevent the lung from, getting this, dead tissue material. And this is what we're trying to do. So we're trying to look for molecules that we can use to prevent this destruction of the tissue. Because if we can prevent the destruction of the tissue, then we can improve the access of the drugs to the places where the bacteria is hiding, and then the drugs will be able to kill the bacteria. So that's what we do with the TB work, is we're trying to understand what drives the tissue pathology, and then look for drugs that can help us to minimize that destruction of the tissue, so that the drugs can get into the tissue and kill the bacteria. So that's the work I do in the main, is I work with parasites and I work with TB. But also my background now is a combination of biochemistry and immunology. So immunology is a study of the immune system. As I said again, when I was thinking about TB, is that, or even when I was talking about the parasites, is the immune response basically is what is key in trying to protect us from infections. So as soon as there's a foreign material coming into our system, the body will react by sending out what we call amasosha omzimba. These are basically soldiers of the immune system that will go into that area of infection, and they will try to attack the infection, whether it's a bacteria or a virus, right? And this is critical, again, to understand in the context of what we are dealing with now, which is COVID-19, where we have this virus that we inhale, and then it goes into the lungs, and it causes problems. Is that ultimately the body needs to respond to this infection. And there are various mechanisms that the body uses to attack this foreign material, right? As I said earlier, is that it will send the soldiers, amasosha, to go and attack. Now, some of these, infections, like a virus or bacteria, can resist that attack, that first line of attack. And then now we need to send reinforcements. Then we send more cells or different types of cells to come in and, respond to this infection. Now, this is what we call adapting to the infection. It's what is called in immunology an adaptive immune response, right? So we have what is called an innate immune response and an adaptive immune response. So in the innate immune response, this is where you find the soldiers, right? These are cells like, for instance, your macrophages, your dendritic cells, your natural killer cells. So these cells have innate abilities of killing pathogens, killing bacteria. So they'll eat it up and chop it up until you have no bacteria left. And then if these cells fail to do their job, and then now you're getting these cells called the adaptive immune cells. I'm sure sometime in your careers, you guys will learn about what is called your T lymphocytes or T cells. Now they are part of adapting. So the cells of the innate immune system, like your dendritic cells, will educate the T cell to say, hey, T cell, we need your help. We have an infection that we're dealing with, but we don't seem to succeed. Can you come and help us? Then they'll show the T cell a bit of the bacteria or the virus. They say, okay, this is what we are dealing with. This is what we are seeing as attacking us. Can you help us respond to this? And then when that happens, then T cells will then go to where there's an infection. And then T cells intervene by secreting what is called cytokines. Cytokines are signaling molecules that will then help to make your cells of the innate system stronger at fighting the pathogen or the infection. And this is how basically these different arms of the immune system work together to kill the infection or the bacteria or the virus or the parasite or whatever that could be causing disease in our body. Now, when we know that this is how the immune system works, and the immune system has these different arms, and I'm not trying to be exhaustive in describing the different arms, I'm just mentioning the two common ones. Of course, it's more complicated than what I'm saying, but I don't think you guys want to know the details now, just as an introduction to how the immune system functions. Now, we can, prevent infections by giving vaccines, right? And this is why now, if you think of COVID-19, we hear now a lot in the media, people are talking about vaccination, right? So the question is, what is vaccination? So if you remember now what I was describing between the interaction between the cells of the innate immune system and a T cell. And I said that these cells of the innate immune system, because they are able to eat the bacteria and take it inside the cell, they then kill it, and then take the parts of the bacteria and then present it to the T cell. So the T cell will know how to respond to this material that belongs to this foreign pathogen, right? This is how exactly what we're trying to emulate with vaccination. We are trying to educate our immune system to respond better to the pathogen when we do get a real infection, right? So we then basically what we'll say, we do a mock infection by either giving the same pathogen, but then we try to inactivate it so that it does not cause disease, right? So you'll see people will speak about heat-killed bacteria, right? So where you take the bacteria, you expose it to high heat, and then basically you make it inactive. So now it can no longer divide. So when you inject that bacteria that is now weak, it will get into the immune system or into our system, but then it will not make us sick. This then allows the cells of the immune system to come in and attack this bacteria that is weak, and then develop the immune system, what we call in immunology, you develop memory to the infecting agent, right? So in future, when the real deal, the active bacteria comes and causes an infection, because now you've seen the same thing before, you are able to have a more robust, a quick response to the real deal now. And then you're able to quickly come in, say, we know you because we have seen you before, and kill the bacteria or the virus. And this is how vaccines work. So as I said, that we have different types of vaccines, and I'm going to speak mainly about 3 different types. So I said we have what you call your whole cell vaccine, and this is where you're taking the full cell. This is either a bacteria or a virus. You then inactivate either through a chemical or through heat or any other way of trying to make this particular bacteria or virus inactive. And then that's basically one form of a vaccine. It's a whole cell vaccine. And then you get what is called a subunit vaccine. A subunit vaccine, basically, this is where in the lab, we will study the virus or the bacteria. We will get the proteins or the molecules that are seen by the cells of our immune system. We will then go in the lab and try and make these same molecules, these same proteins, and then we use these proteins as a vaccine to try and educate our immune system, right? So we're using a part of the bacteria or the virus to educate our immune system. Now, the last type of vaccine I want to talk about is what is called an mRNA vaccine. And again, you probably have seen this with the COVID vaccines. Is that, for instance, there are 2 companies currently that are making mRNA vaccines. It's Pfizer and a company in the US called Moderna. So mRNA basically is, if you think about how do we make proteins in our body. So first is we have DNA. So DNA, this is where we have all our genetic information is contained in our DNA. Now, if we want to make proteins, we then take this DNA that we have in our cells, and then it goes through a process called transcription, right? So we transcribe this DNA into RNA, right? So we're converting all the information that is stored in the DNA into this RNA molecule. Now the RNA will undergo a process called translation. All of this happens in our cell. It's completely automatic. You don't even, as I'm speaking right now, these things are happening in my body. There's DNA that is being read, there's RNA that is being translated into proteins. And proteins, as I said earlier, they are the effector molecules, so they perform the functions in our body, like signaling, sending a signal from one cell to another cell. Like now, I'm recording, I'm speaking. There are muscles moving, there are proteins moving. All of that happens automatically. I don't think about those things, they just happen. The brain is able to process and coordinate all these signals, right? So once you have the mRNA, then the mRNA can then be translated into the proteins, right? Which in this case, in terms of COVID-19, they are targeting one protein in the main, which is called the spike protein. So if you look at the structure of COVID-19, on the surface we have these spikes, right, that are on the surface of the virus. And it is these spikes that are on the surface of the virus that are seen by the cells of the immune system, right? So now we can go to the DNA or the RNA. The RNA can, give instruction to make these spike proteins, right? And this is what they do with these vaccines. That this mRNA that they have in the vaccine has information for the synthesis of the spike protein. And then once we have the spike protein, then our immune system will say, oh, hey, what is this thing that is in our body? This is foreign. Then they will take it up and then build memory to say, this is foreign. In future, if we see this protein again, we need to respond to it. And then so when you then get the real infection with the coronavirus, because the spike protein is the same, then our immune system is more stronger to deal with the real infection. And this is how then we are able to overcome the infection. And basically, in terms of COVID-19, we have these 3 different types of vaccines available. Now, you guys will be asking yourself, because, again, people are talking in the media, but these vaccines are not safe, but these vaccines are going to, we are injecting ourselves with 666 and all those theories that are out there, right? Now, I hope that I've been able to clarify that how vaccines are synthesized in the lab and what they're supposed to do. They're supposed to basically, prime your immune system so that it's able to respond better to the real deal, right? And this is what they do. Now, in South Africa, we had, learned that we had the AstraZeneca vaccine. Now, again, the AstraZeneca vaccine, I would say that basically is based on what is called a vector. So a vector basically is something to use to transport something into our cells, for instance. But in this case, they use what is called an adenovirus. So it's a type of virus, right? In the same way that HIV is a virus, in the same way that coronavirus is a virus. And then in this virus, what you do is you encode the spike protein, right? So basically, it's a vehicle to deliver the spike protein to our cells, and then we then make the spike protein, which then educates the cells of our immune system. We develop immune memory. Then when we get the real thing, we are able to deal with it, right? Now, when we got this AstraZeneca vaccine, it was made specifically to target a specific type of spike protein. Now, again, because the virus is existing under this immune pressure, and the aim of the virus is to survive in our system, it's not to die, right? So when the immune system comes and attacks the virus will respond by mutating itself, as I mentioned earlier with TB. This is how we get, drug-resistant TB, is that the bacteria will change a bit of its DNA so that it's able to resist the attack. And it's the same thing with COVID-19, is that as the virus is dividing, it's multiplying, there's going to be changes introduced into its genetic information that will allow it to survive attacks from the immune system. And this is what happened with the COVID-19 vaccine, where the one from AstraZeneca was found to be ineffective against our variant in South Africa. because this variant had undergone mutations in the spike protein that allowed it to basically evade the immune system. And this happens all the time. We see that also with HIV. So it's not only unique to coronavirus. HIV does the same. The reason why today we don't have a vaccine for HIV is because of the high rate of mutation of the virus. In fact, viruses are known to be, to undergo a lot of mutation. So it's very difficult to develop vaccines for viruses because of this mutation. And again, just maybe to put things into context, is think about influenza. Every year, you are supposed to take a flu vaccine. So you don't take one flu vaccine and assume that you're going to be protected for 10 years, right? Because influenza is a virus undergo mutations. So every year, the flu epidemic is driven by the different variant of the flu virus. Hence why every year we are supposed to go and get a new flu vaccine. And probably it's going to be the same thing with coronavirus. It's because when you look at how fast the virus is able to mutate itself, right? I mean, we've had a variant in South Africa. We've heard of variants in the US. There have been variants discovered in the UK. There have been variants discovered in Italy, right? So it shows you that the virus is trying to survive in our body, is trying to survive the attack, coming from our immune system, and it's then undergoing all these mutations to try and survive. So it may be that with COVID-19 as well, we may need to take a vaccine every year. We don't know, right? Studies are ongoing. People are doing the research and they're monitoring the situation to say what is happening. How long does the vaccine-induced response remain in the system? Can we get protection, after 1 year, 2 years, 5 years? All that information is still not known because this is a new epidemic that we're dealing with, right? It's only from last year. And we can say it's only really been from this year where we've seen a mass rollout of vaccination all over the world, right? I think as things stand, we have just above 5 million people who have been vaccinated so far. So you can see, how fast we are going. And I think every day now, I think in the US, they're really trying to ramp up the vaccination efforts. And these numbers are going to balloon as more vaccines become available. So in South Africa right now, what we have is, so they had to discontinue the AstraZeneca vaccine because we had a different variant and it was shown to be ineffective against, protecting against mild and severe disease in patients. So the people who took that vaccine still developed mild and severe symptoms. So it did not reduce those severe symptoms. So it was like, what's the point of using that vaccine if we cannot even reduce the need for hospitalization in our patients? And then now we have what is called the J&J vaccine. The J&J vaccine also, in terms of how it's made, is similar to the AstraZeneca vaccine. But at least in our context, it's shown to be effective against our variant. This is a 541 variant that we have in the country. So that's the promising news. But what we have currently in the country is we don't really have a vaccine rollout plan, right? But what we have is a study that is ongoing. And they've been trying in the first phase to vaccinate frontline healthcare workers. So this is basically part of a study. This is to get more data on the efficacy of this vaccine in our setting. And then, as we get more doses, then they'll move from a study now to a mass rollout for everyone in the country. But that's going to take time. Probably, if you think about it, from where we are now to the point where we are able to vaccinate the majority of the country, maybe looking at 2-3 years. Now, again, there are a number of questions that you can think about, right? Is to say, by the time that we have mass vaccination of the population, what variants are going to be in the country? And will the vaccines that we have now be effective against those variants, right? I don't know the answer. No one knows the answer. Only time will tell. But as things stand, we only have access to J&J vaccines, and they are being rolled out as part of a study done by the guys at the MRC. So this is where we are as a country in terms of COVID-19 vaccines. Now, the question is, are vaccines safe? And should we take vaccines? Because I think everyone has been talking about that. And I want to assure you guys that all of us in this country have taken a vaccine at some point in our life. I'm sure some of you guys have seen colleagues of yours who have, dots on the right or left arm. This is because of a vaccine, specifically a TB vaccine that we all get given when we are young. So in South Africa, every child has to undergo a vaccination program. And I think you take vaccines up to the age of 12, if I'm not mistaken. So there are vaccines that you get from birth. As soon as you're born, whilst you're still in the hospital, there are vaccines that you receive at that point. And then you need to go in, I think, after a year, after 2 years, and so on and so forth. So vaccines are normal. And in fact, some of the, infections that were problematic in the past, we've been able to eradicate them or reduce them because of vaccination, right? A good example I can give, for instance, is polio. The reason why we don't have polio is no longer in common today is because of mass vaccination. We were able to, reduce the impact of polio in our populations because of vaccination. So actually, vaccines are a good thing in terms of controlling disease at population level. You can think about, for instance, TB, right? Is that we have a TB vaccine. In this country, everyone gets a TB vaccine. It's called BCG. The full name is Bacillus calmette-guerin, right? It's BCG. And basically, it's derived from a variant of the Mycobacterium that infects animals, cows in particular. So they were able to make this strain of bacteria weak and then use it to inoculate human beings to give protection against Mycobacterium tuberculosis later on. And this vaccine is very effective, particularly at a young age. Then as you get older, the efficacy of the vaccine reduces. But as an infant, when you are young, the vaccine is very, very efficient. So in my view, and based on my knowledge and understanding, I think vaccines are very important in terms of dealing with disease, particularly in the context of a pandemic, where one is we need to balance the need of people going to work and earn a living, and two, balance the health of the population, right? And somehow people need to live, but people need to remain healthy. So how do we find a balance? And I think a vaccination program will help us to at least know that people can be protected against the virus in future. So that's really what I wanted to discuss with you guys today. You'll see that, I've touched on where I come from, which is a village, and moving from the village, getting into university, which is mainly was just by luck. And again, maybe it's being born at the right time and going to university when the demand was not so great. I told you a little bit about what I do at UCT, which is what I do with parasites, what I do on TB, trying to reduce the pathology. I've spoken to you about vaccines, what are vaccines and why we need them, particularly in the context of COVID-19. And I hope that you guys will be able to find something that is of interest to you from this talk, and you are inspired in some way to look for careers within the science field, because we need more young black people to enter the space, the biotech space. There's more room for innovation. There's more room for research. If you look at the number of diseases we have in Africa, some of them are completely under-researched. So we need more young people to come in and take on these problems and try and find solutions. And for you to come and do a degree in science, you need to do well in mathematics. That's number one, right? And you need to do like proper maths, not maths literacy. That's one thing. So if you know that you have a desire, for instance, to be a doctor or to be an engineer or to be a scientist, already now when you select your subjects at grade 10, you know that you need to do maths, right? And if you want to get to university, because now you're competing for space, you need to get at least, I would say a C. What is a C now in levels? I think it's a level 5, I think. Because when I did matric many years ago, we had, standard grade and higher grade, and we had the different symbols, A, B, C, D, E, F, G, H, sometimes. But then now things have changed. We have levels, level 7 to level 1. So I think a C is a level 5. So that's really should be your minimum, really. And if you want to get to universities like UCT, VIS, and so on, then really you need to be on top of your class, really. That means you need to get 80, 85 to get a space in these universities. So the sooner you decide what you want to do, the sooner you can put in the work to achieve that goal, right? There's no point in you saying you want to be an engineer, but you start really falling in love with your work at matric. It's too late. And subjects like maths, like physics, they require repetition, right? And as a teacher, I always tell my students that repetition is a mother of learning. You can only learn by doing something over and over and over again until you get the understanding in your head that, okay, when I do algebra, this is how I approach it. When I do trigonometry, this is how I approach it. This is why this theorem of Pythagoras makes sense and how to apply it in this particular, context. So you need to decide early on and then follow through with the practice every day, Monday to Sunday, until you get to matric, until you write your exams. And probably when you get to university, you need to do 10 times more than what you were doing in matric. So for sciences, you're doing your maths, you're doing your physics, and I think you have to do your biology as well. Then you do well in your languages, your first language and your second language. Then if you do well in those subjects, you have good grade 11 marks. Grade 11 marks get you seen by the university. They give you a conditional offer. A conditional offer means basically we like you. Now we just need to see your final marks, we'll give you a space. Right? And most people don't even get to the stage. So at the end of the year, when they get their matric result, they're busy calling universities. The university does not even know who you are because your grade 11 marks were so poor that they chucked away all your application. Like, we're not even going to bother to look at you. Because how many people are applying to get to these universities? They can't look at 26,000 applications only to give 5,000 spaces for first year students for the whole university, from commerce to humanities to engineering to medicine. So they need to have a strategy on how to get the best people and then give the small pool of people the best analysis and look at those closely. So you want to have that opportunity to be looked at. And it requires you to do the hard work early on in grade 10, in grade 11. By the time you get to matric, you're just consolidating the work that you've done at grade 10, 11. So that's the first part. The second part I would want to leave you guys with is, just knowing exactly what you want is critical. Because once you know what you want as a high school learner, then you're able to navigate the peer pressures of being a young person. You are able to sacrifice if you have to sacrifice because you know exactly where you want to get to. Now the problem comes when we allow friends to influence our decisions and influence our behavior. And I can tell you right now is we don't pass as a collective. Also, you don't fail as a collective. So you need to be clear about your ambition, your goals, and dedicate time and effort to achieve those goals. No one will do it for you. No one will do it with you. But you can find people who have similar goals and you guys can work collaboratively once you've defined exactly what you want. There's a book I read. It has a very nice title. The book was written by Paulo Coelho. The title says that winners stand alone, right? To say that if you want to win, you don't look around you and say how many people are there with you, right? Because you are the only one who has those dreams. unfortunately, right? You're the only one who can see that this is a problem that is there in my community and I want to solve it. Let's say you are an entrepreneur. You see a problem, you want to solve it. You're the only person who sees that vision, right? So now if you're going to say, well, I can't start solving this problem until I see my friends also having an interest in the same problem, that means you're never going to get anything done. Hence why this book says that winners stand alone. It's only when you're on the other side and you have won that people want to come and join in. But in the struggle of you trying to win, more often than not, you're isolated. And I think this was true for myself as well. Where, for instance, is, doing, undergraduates, doing postgraduates, doing a postdoc, I was mainly by myself. Because my friends, maybe they did their undergrad degrees, they finished, they went to work. They wanted to have a job and an income. I was still a student. So if I looked at my friends and say, well, my friends now are driving cars and I don't have a car, and I allow the pressure to dictate how I should live my life, I would not be here today talking to you guys, and now you can call me Dr. Hlumani Ndlovu, right? I would still be Hlumani Ndlovu somewhere in a company working every day, but now I have all the freedom, right? Now, I can choose where to go, when I want to go, and all the stuff. Even with the work I do, no one really sits on my back and say, Hlumani, have you done X and Y and Z? I have so much freedom because of the education I received. And this education has been able to give me opportunities. For instance, I've been able to travel overseas for free without paying a single cent. You're able to go there, someone will pay you for your hotel accommodation, someone pays for your food, someone pays for your flight for free. Why? Because I've been able to differentiate myself from, my peers. I've been able to get this education that says that I'm different. I'm able to do the work that I do, that some people find it interesting. They say, come and talk to us about your work. And this is the prize of winning. Of course, when I was alone, spending 10, 12 hours a day in the lab, people will laugh and say, oh, look at him, he can't even afford to buy a new pair of shoes. Because, I'm living on a stipend as a student. But when the outcomes, at the end, the rewards come, for me, I find it to be worth it, ultimately. But also what for me is a plus is now I have an opportunity to work with young people as an educator, as a lecturer. And I'm able to transfer my knowledge to them, and I'm able to inspire them. And these are young black people, in the majority, who will go into our public healthcare system and treat our parents who don't have medical aid, right? So now I'm able to give back through my knowledge to the country and train the next generation of doctors who are going to have an impact in the country. And I think that is fulfilling as well. Beyond the money that you get paid at the end of the month to say that you are a lecturer at UCT, is the fact that you are able to have an impact on young people who are going to have an impact on the country, right? And I think that is very, very beautiful to have the opportunity to impact, young people in that way. So that really is my motivation to a young person right now who is in high school, who's unsure where to go, who's unsure what they want to do. I'm like, spend time, talk to your teachers, ask questions, go to expos when they are advertised, and go and meet people from different careers. Learn what does an engineer do, what does a scientist do, what does a doctor do, and find yourself or find your interest within those different careers, and then apply yourself every day to achieve that goal. And I can assure you now is if you put in the effort, there is no way you won't be rewarded, regardless of how poor you are, regardless of how the environment you come from. If you do the work, I can assure you, people will come to you and say, we've seen your hard work, here is a bursary, go to UCT. Here is a scholarship, go to Harvard. Here is a scholarship, go to Oxford, because you have risen above your prevailing circumstances and you have shown yourself to be capable of succeeding anywhere in the world. And this is what I wish for you guys, is to have the ambition and the drive to achieve a level of success that will take you anywhere in the world, depending on your desires. So thank you for listening to me. I will make myself available to talk to you guys when the video is being aired, and you guys can ask me any questions you have about my work, about my education, about where I come from. Thank you for the opportunity. Sharp, sharp. Was it good?